So week 14 focuses on the kingdom Animalia. Here's a list of the learning objectives. We're going to start by going through an overview of animals. So animals are diverse, yet they all have features in common. They have multicellular bodies, eukaryotic cells with no cell walls. They're heterotrophic by ingestion. Heterotrophic means that they eat other organisms in order to survive. They have a blastula, or a hollow ball, stage of development, and their cells bind to extracellular matrix. Most animals are invertebrates, which lack backbones. A scorpion is an invertebrate. Much fewer animals are vertebrates, which have backbones. However, vertebrates may be more familiar to us since they include mammals, amphibians, reptiles, and fish. So when we think of an animal, we usually don't think of an insect. We think of like a puppy or a kitten or something like that. But, a majority of animals on Earth are actually invertebrates. The first animals arose from aquatic protists about 570 million years ago. Many animal phyla originated during the Cambrian explosion. And these are the 9 major phyla of animals. We will be talking about the major characteristics of all of these 9 phyla. As life was evolving, the first branching point in animal taxonomy distinguishes a clade with true tissues from a clade with no true tissues. Parazoans, sponges, cells do not interact to provide specific functions, so sponges have no true tissues. Eumetazoans do have true tissues. The second branching point distinguishes radial symmetry from bilateral symmetry. An organism has radial symmetry if any plane passing through its body from the mouth to the opposite end creates mirror images. An organism has bilateral symmetry if only one plane can divide the animal into mirror images. Bilaterally symmetrical animals have a head and a tail end. This body plane selects for cephalization. Sensory organs and a brain are on the head end. The same branching point also distinguishes animals with the 2 embryonic germ layers from animals with 3. In all phyla except for sponges, the embryonic ball of cells, called a blastula, folds in on itself, forming a gastrula. In some animals, the gastrula only develops 2 true tissue layers, endoderm and ectoderm. In others, a 3rd tissue layer develops, called the mesoderm. Ectoderm develops into skin and nervous system, endoderm becomes the digestive tract, and the mesoderm gives rise to the muscles and circulatory system. Embryonic development also determines the 3rd branching point. In the first indention of the gastrula develops into the mouth, the organism is a protostome. If it develops into the anus, the organism is a deuterostome. The body cavity, organization of the digestive tract, and segmentation also characterize animals. A bilaterally symmetrical animal may have a coelom, a pseudocoelom, or no coelom. A coelom is a body cavity surrounded on all sides by mesoderm. A pseudocoelom is surrounded on one side by mesoderm and the other side by endoderm. Some bilaterally symmetrical animals lack a body cavity. Animals have an incomplete digestive tract if the mouth both takes in food and ejects wastes. Animals have a complete digestive tract if food passes in one direction from mouth to anus. Some animals have segmentation, like this millipede. Segmented bodies are more flexible and have more potential for developing specialized body parts than unsegmented bodies. Animals are also classified by the presence or absence of a larval stage. Animals with direct development resemble adults in their juvenile stage. Animals with indirect development have a larval stage that does not resemble the adult form. The larva undergoes metamorphosis as it matures into an adult. Different combinations of these qualities characterize each phylum.